out, and then making a tadpole. And here then is the picture, which you might not find as attractive as many kind of Jiminy Cricket frogs, but it's a beautiful frog to many of us because it's the first adult cloned animal. So on the top is the donor from which an intestinal cell was removed, and on the bottom is the first clone, in 1962 by John Gurdon. Now this technique then, which I'll describe in more detail later, shows that it's possible to make a genetic copy of an adult animal through this nuclear transfer. And here's an example where we're going to look at many copies. So on the left you see the egg donor, so in this case the female frog, which is, I might note, about 40% by weight ovary, an animal pretty much devoted to making eggs. Many eggs were removed from her, and then they were transplanted, that is, nuclear transfer was accomplished using a blastula from a mating of the albino frog shown in the middle. And those produced then these 30 identical, that is, genetically identical cloned adults on the right. You can think of this like sets of identical twins, but they're genetically identical. One of the points I'd like to make here is if I could draw your attention to what is here, the third row, and if you look at like the third and the fifth frog over, those frogs are smaller than the other ones. That's because they probably didn't compete as well for food or didn't do as much exercise. And this should just remind you of something I think you already know intuitively, is that your genes are not your destiny. They're important for setting limits on what can be done, but what happens in your life can make a big difference. So here we have 30 identical frogs, but they didn't all grow up to look exactly alike. And if they did something more interesting than they normally do, like if they could listen to music or think, we could test whether or not they actually had different abilities, but can't do that with a frog. Now, many of you will have heard, of course, since cloning is in the news so much, that cloning frogs has not really captured the public's attention. It was cloning mammals that got everyone's attention. So here is a picture of maybe one of the most famous clones, the sheep Dolly. Dolly was cloned from a mammary epithelial cell, and then the nuclear transferred embryo was put into a foster mother, shown here on the right, the black-faced ewe, and she then gave birth to this clone, the sheep Dolly on the left. Dolly grew up a year or so later, and then became a mother herself, making her little baby Bonnie. Now, there are a number of animals that have been cloned. Sheep aren't the only ones, and I thought I would show you a video of how cloning occurs. So we're now going to look at the mechanism of it. And before we start the video, I thought I would just say that one of the things that we want to attend to here is the pipettes, one on the left and one on the right. This video is prepared by my colleagues Dieter Egli and Kevin Egan. And you see here on the left is what's called a holding pipette, which gently sucks on the egg. The egg is surrounded by a membrane called the zona. And you'll watch this pipette on the right first drill a hole into the zona, then go in and suck the nucleus out. And then another nucleus, which has been taken from, say, a somatic cell, a cell of the body, is going to be put in. So if we could start the video, please. You'll see now that this drilling pipette is going to drill a little hole into the membrane. You can maybe see a little bit of the hole right here at the next part. This pipette, you'll note, isn't really sharp like a syringe. There you can see a bit of the hole. And now the pipette's going to go in and remove the nucleus. And if you look carefully in the pipette, you'll see a line in the nucleus, which are all the chromosomes lined up. So that nucleus is going to be squirted out now because we don't need it anymore. And there we have an enucleated egg. Now the next step is to take a set of eggs like that, and I'll show you two, and then transfer into them a nucleus from another kind of cell, a fully differentiated somatic cell. So here the enucleated egg is set on the side, that it's held by this holding pipette on the left. There's drilling the little hole in the membrane. Here we go in. Here comes the nucleus from the right. The pipette goes in, and these pipettes are operated with a piezoelectric device. So you can't see it here, but it's like a little jackhammer going very quickly, like a Woody the Woodpecker, getting in there to then squirt the nucleus in. Here we'll see it again, a little hole, and the zona is prepared, and now the nucleus is going to be squirted inside. So there's two examples of what's called somatic cell nuclear transfer. So that's a sort of long way of describing cloning.